In my lab, we are trying to identify cancer, lung and pancreatic cancer vulnerabilities. And in particular, we are looking at we are studying the tumor biology of these tumors in order to identify signaling pathways that could be targeted and then later on develop drugs to inhibit in the clinic in human patients to block important signaling pathways that promote the growth and development of cancer. So I'm born and raised in Greece, and I studied in Italy. I did my master and a bachelor studies in Italy. I studied biology, and later on I moved to the US for a postdoc, and then in Geneva for another postdoc, and then I won the Swiss National Science Foundation professorship, and I established my lab here since 2016. I'm as a professor, and I have a small group here at the Institute of Pharmacology. We work in vitro using human lung and pancreatic cancer cell lines derived from human patients. And in vivo, we use state-of-the-art mouse models of lung and pancreatic cancer that very closely recapitulate human disease. And we also, of course, combine our observations with collaborations with the surgeons and pathologists where we gather some human samples. One of the important aspects of University of Bern is our infrastructure. And in particular, we have recently purchased an imaging mass cytometer, which with a cooperative work from the Inselspital and the University of Bern, and it's actually situated in our institute here. And this technology gives us a lot of information now. With this imaging mass cytometry, we can actually stain for multiple markers, up to 30 or 40 antibodies, different antibodies. So especially for our type of research, lung and pancreatic cancer, we have to be able to understand not only what is going on in tumors, but also in the tumor microenvironment. And here it's an example. You can see a multiple staining. In these specific samples, we did a 10 marker staining, and here you can see them separately now, but this is basically the same image. We studied this signaling pathway, we identified key molecules of this signaling pathway, and then when we targeted with pharmacological inhibitors, we could suppress tumorigenesis. We could boost the immune system to fight pancreatic cancer cells, and importantly, we could limit the metastasis formation to the lungs. It requires a lot of commitment because we work long hours, we work weekends and even holidays, so especially because we are also working with mouse models and they require a lot of work. And however, I would highly recommend to new researchers if they want to pursue this type of academic career, because there is not only bad side, but the good side is that one can really make a change in the treatment of, for example, in our case of lung and pancreatic cancer. We have the freedom to choose our projects and pursue important questions to fight cancer.